We'll continue talking about the signaling. So in endocrine system, generally there are three modes of signaling: the paracrine signaling, synaptic signaling, and the endocrine signaling. So actually the paracrine signaling, when the cell secrete a signaling molecule that affects a target cell that's nearby the cell. So these signals are called local mediators. It only affects cells nearby. That's a paracrine signaling. While synaptic signaling occur only in the nervous system, where the nervous cell sits by its axon, the long axons and the end of the axons, on a target cell. This target cell is either a gland or a muscle. And the signaling molecule here in this case is called neurotransmitter. While the endocrine system is totally different, in which the endocrine gland secrete the signaling molecule that in this case is called hormone into bloodstream. And this hormone travels along the bloodstream until reaching to its target cell or target organ. So you can notice that the slowest signaling is the endocrine signaling. But the nervous cell transmits information by impulses in a speed of 100 meters per second. These neurotransmitters that come from the nerve terminal diffuse in a distance around 100 nanometers to the target cell. So the main difference here that the hormones are greatly diluted in bloodstream, while the neurotransmitters are less diluted and can achieve high local concentrations at the target cell. Once the neurotransmitter is released from the nerve, it must bind to a receptor that must have a low affinity for the ligand or ligand, which means that the neurotransmitter can dissociate rapidly from receptor to terminate a response. And this is obvious actually. This neurotransmitter need to have low affinity with its receptor. Otherwise, the effect of the neurotransmitter will last for a very long time. So it needs to bind to the receptor, do the action, activate the target cell, and got released or got degraded this neurotransmitter. So it must have a low affinity to its receptor on the target cell. So you can see here, for the neurons, it's a kind of a very specific signaling. So you can see that three different neurons, A and B and C, each one of them can sit on a different cell. So it's a kind like one-way transmission. Each neuron will give a specific signal for a specific target organ. While the case, although it is specific in the endocrine as well, but it needs the bloodstream, and it is transported along the bloodstream until reaching its target organ, and needs as well a receptor. So from here you can see obviously that the signal transmitted via the endocrine system is highly diluted since it moves in the blood, while for the neurons is highly concentrated because it travels a very small distance. We have said before that around 100 nanometers. Actually, it is the distance between the end of the neuron and the target cell. How the decision of groups of cells do the same decision by to have a final decision. How does that occur? Cells can send signals to themselves. That's as we said before, that's a paracrine signaling, or other cells of the same type. You can see that during development, for example, the autocrine signaling. What's meaning autocrine signaling? That means the cell signal itself to undergo development. This autocrine signaling is used to encourage group of identical cells to make same developmental decision. We can see here in the first figure, you can see that a single cell can secrete these signaling molecules, which will have effect autocrine on itself or either a paracrine, which is paracrine to the nearby cell. So in this case, what's the difference between the two figures? The first figure that shows that initially a cell secretes a signal that activates itself. Once the signal activates the nearby cell, it will enhance these nearby cells or neighbor cells to autocrine secrete and activate itself as well. So that means this enlarges the signaling occur, and this will lead that all these group of cells will have the same decision of action or development. And this scenario is very crucial in the developmental process. Generally, there are 4 methods of signaling, ranging from the direct to remote communication. The first one is a direct one, in which the signaling is either ions or small molecules, and there is a direct movement through gap junction. That means there is a signaling between 2 adjacent cells that is already attached to each other, and these signaling molecules move through gap junction between these 2 cells. And the perfect example of that is the ions flowing between the cardiac muscle of the heart. While the synaptic signaling, as we said, the type of the chemical messenger here is a neurotransmitter. And this neurotransmitter diffuses from neuron across a very narrow space, as we said before, 100 nanometer, which is called the synaptic cleft, to the cell bearing the receptor, which is either a gland or a muscle. And the famous example of the neurotransmitter is acetylcholine. The third one is the paracrine signal. As we said, it's a local hormones, so it affects the nearby cells. The diffusion occurs through the extracellular fluid to the nearby cells and attached to the correct receptors. An example of that is prostaglandins hormones. Prostaglandins chemical compound or chemical messengers. For the endocrine signal, it's the only type of signaling that needs the bloodstream for being distributed. Hormones is a perfect example, and the insulin is a famous hormone as an example. And this hormone needs to be carried by the bloodstream to the nearby or distant cells having the correct receptor. So how hormones are classified? Basically, they are classified according to 3 criteria. According to the anatomical source, according to chemical composition, according to solubility. For the anatomical source, you can see that's mean according to the source of the hormone, where the hormone came from. The first gland is the hypothalamus. The hypothalamus secretes a number of important hormones that trigger the release of other hormones, as we will see later in lectures. So hypothalamus secretes growth hormone releasing hormone, growth hormone inhibiting hormone, or somatostatin, GHIH, gonadotropin releasing hormone, corticotropin releasing hormone, thyrotropin releasing hormone, prolactin releasing factor, as well as prolactin inhibiting hormone, PIH. As you can see, most of the hormones of the hypothalamus actually they are either releasing or inhibiting. So that means the hypothalamus is a very important gland that might control another gland, as we will see in the next slide, to release its hormone. So, for example, if the hypothalamus release growth hormone releasing hormone, that means this hormone will trigger another gland, which is the pituitary gland, to release the growth hormone. While if the hypothalamus release growth hormone inhibiting hormone, it will trigger the gland to release, it will trigger the negative feedback mechanism to stop the production of the growth hormone. So this is the first gland to secrete hormone. The second gland is the pituitary gland. Actually, pituitary gland is composed of two regions, the anterior and the posterior. Both of these two regions have specific hormones. The first one is the anterior pituitary gland. The first hormone is the corticotropin hormone or adrenocorticotropic hormone, the thyroid stimulating hormone, and it's obvious the thyroid stimulating hormone, which is secreted from the anterior pituitary, have an effect on the thyroid gland to release its thyroid hormones. The next hormone is the melanocyte stimulating hormone, growth hormone, it's a major hormone in our body, or somatotropin, prolactin, gonadotropin hormone like the FSH, follicle stimulating hormone, and the luteinizing hormone. While the posterior pituitary gland secretes only two hormones, which are actually very important hormones, which is the antidiuretic hormone or vasopressin, and this hormone is very important in water retention inside our body. So when our body feels that it is less in water content, the posterior pituitary secretes antidiuretic hormone to prevent further loss of water from our body. While the other hormone from the posterior pituitary gland is oxytocin. And oxytocin is a very important hormone during labor for the delivery of babies, as we will see in the coming lectures. So only posterior pituitary gland secrete two hormones, the ADH and oxytocin. Continuing according to anatomical structure of the classification of hormone, you will find the thyroid gland secrete thyroid hormone, T3 and T4, as well secrete the calcitonin, which regulate the amount of calcium in our body. As well, it secretes the parathyroid gland secrete the parathyroid hormone. While the adrenal gland has two regions, the adrenal cortex and adrenal medulla. The adrenal cortex is responsible for secreting of the steroid hormone. And you need to know that the steroid hormone, it is the only kind of hormone that doesn't need a receptor on the surface of the target cell. Because the steroid hormone are lipophilic, so they can easily penetrate the cell membrane. So you can see the examples of the steroid hormones like the cortisol, aldosterone, and DHEA. While the adrenal medulla secrete two of the most famous hormones is the adrenaline and noradrenaline. Adrenaline, which is epinephrine, and noradrenaline is the norepinephrine. The kidney secrete four hormones: the erythropoietin, 1,25-dihydroxyvitamin D3, renin, and angiotensin 1, important for the function of kidney. The pancreas, of course, secrete two major hormones, the insulin and the antagonistic hormone to the insulin, which is the glucagon. While the gonads secretes the sex hormones, as example, the estrogen, progesterone, testosterone. So in the last two slides, we saw how the hormone is classified according to anatomical structure. Now we'll talk how the hormones are classified according to chemical composition. Hormone can be peptide hormone. What's a peptide hormone? Actually, it's a hormone that consists of amino acids. It's not a huge protein. It's a peptide hormone. You can see the types is a polypeptide hormone, like the hormones that come from the anterior pituitary glands, like the hormones that come from the placenta in the mother, in the pregnant mother, and these placental hormones are anterior pituitary-like hormones, as well as the pancreatic hormones. The parathyroid hormone and the calcitonin are polypeptide hormone, as well as erythropoietin and renin coming from the kidney are polypeptide hormones. For the oligopeptide hormones, They are secreted from both hormones that come from the posterior and pituitary gland, which is the ADH, antidiuretic hormone, and oxytocin. They are oligopeptide hormones, as well as the hypothalamus hormones, except the PIH. What's PIH? It's the prolactin inhibiting hormone. So if we see here how these polypeptide hormones are synthesized inside the cell. Of course, it's a protein in nature. So you have the DNA and you have the gene for that hormone. When the RNA polymerase binds to the hormone, it leads to transcription, and you have a messenger RNA precursor or pre-messenger RNA. Upon processing, which is the excision of introns and joining of exons, and another modification in the processing of the messenger RNA, the messenger RNA become mature. This messenger RNA is translocated into the cytoplasm, being covered by a cap at the 5' end, which is called M7G PPP, methyl 7-guanylate triphosphate, in the cap as a cap for protection at the 5' end of the messenger RNA. And you will have a polyadenine tail at the end of the messenger RNA at the 3' end. So the mechanism of maturation of the messenger RNA occur actually on as a 3 different modifications. The first one is the processing, which is the removal of the introns. The second one is the addition of a cap at the 5' end. The third one is the addition of a polyadenine tail at the 3' end. And this process is called post-transcriptional processing. And it occurs for the pre-messenger RNA, which is transcribed from the DNA. And all this process, this post-transcription process, occur inside the nucleus. So the messenger RNA had to leave the nucleus already processed. Once the messenger RNA is mature and have these 3 modifications in the cytoplasm, it comes to the ribosomes, which is connected to the endoplasmic reticulum. And these ribosomes, with the help of the transfer RNA carrying the correct amino acids, start synthesizing of the hormone, the polypeptide hormone. Okay, in the Golgi region, in the endoplasmic reticulum, the pre-pro hormone is formed, which we can call it the precursor of the functional hormone. So it is stored inside the lumen or cisternae of the endoplasmic reticulum. This pre-pro hormone got spliced to form a pro hormone through the process of cleavage, and still in this case is still immature hormone. In the Golgi regions, another modification is added to the hormone, and another sequence of amino acids is cleaved away to form the mature hormone. This mature hormone in the Golgi region got packaged into vesicles and got secreted into the bloodstream after all this modification to be transported to its target organ. So the level of modification of hormone starts from the post-transcription processing, then the modification that occurred through the process from the endoplasmic reticulum until reaching the Golgi region, until it's reaching its final destination to do its action. So you can see here again, this is the rough endoplasmic reticulum. You can see it occurs first, the translation in the rough endoplasmic reticulum, then the translated polypeptide hormone is transported into the Golgi region, where in the Golgi region there is a lot of modification occur, and as well as there is a sorting mechanism for hormone. And these Golgi apparatus or Golgi region have the ability to package the final sort and package the final hormone into secretory vesicles. As you can see, this secretory vesicle either secreted immaturely to the bloodstream or secreted mature secretory granules to the bloodstream. How the cell decide this fate is based upon the function of the hormone. Some hormones need to be secreted as an immature form. Other hormones are secreted as a mature form. So what happens if there is error in this process? What happens? There is an error in the formation of this polypeptide hormone. If there is any error in the sequence of the amino acid, these Golgi apparatus have the ability to sort and isolate this defective polypeptide hormone in a vesicle that got digested by the lysosomes. So what is the importance of this step? The importance that the lysosome digests this defective hormone to reuse its amino acids for making new hormone. Continuing according to chemical structure, so we just have dealt with the polypeptide hormones. The other types of hormone is amine hormones, and these all derived from the amino acid tyrosine. So you have the thyroid hormone, which is the T3 and T4. You have the adrenaline hormone. You have the noradrenaline. You have the prolactin inhibiting hormone, PIH. The third type, according to chemical structure, is the steroid hormone. And all of these hormones are derived from the cholesterol. Like the sex hormone from the gonads and placenta, all the adrenal cortex hormones, and 1,2,125 dihydroxy vitamin D3. So you can see here, as an example, for the peptide and protein hormone, you can see the structure of the oxytocin. That's why it's a polypeptide hormone. It's a pure amino acid. As well here you can see how this hormone, the norepinephrine and the thyroxine, it's an amino acid derivative. It's only synthesized from one or two amino acids. So that's a major difference between these two types of hormone. You can see here that the steroid hormone, as we said before, it is synthesized from the cholesterol. As an example, you can see the testosterone and estrogen. The third category of classification is according to solubility. So majorly we have two kinds of solubility. Either the hormone are water-soluble hormones or fat-soluble hormones. So the water-soluble hormone, of course, all the peptide hormone and all the amine hormones except the thyroid hormone. For the fat-soluble hormone, the steroid hormone, and as I said before, the advantage of the steroid hormone is the ability to move across the cell membrane, and it doesn't need a receptor on the surface of the cell, on the surface of the target cell. Although it doesn't need a receptor on the surface of the target cell, but it still needs a receptor to bind in the cytoplasm. So the steroid hormone have the ability to transverse the cell membrane of the cells, but need to be attached to a cytoplasmic or a nuclear receptor to perform its function. We'll talk now about the mechanism of hormone action. So actually, hormone interact with a receptor to affect the target cell. These hormone receptors are located either on the plasma membrane, as we said before, or inside the nucleus. And by now you know already what's the receptor inside the nucleus. The receptors inside the nucleus are important for the steroid hormones, which can transverse easily the cell membrane. So we have hydrophilic hormone and the hydrophobic hormone. So generally, the hydrophilic hormone acts on plasma membrane receptors, while the hydrophobic hormone acts on nuclear receptors. So we'll talk now about the tropic hormone. What's a tropic hormone? Tropic hormone that stimulates an endocrine gland to grow and secrete its hormone. In human, tropic hormones are secreted by the adrenal hypophysis, which actually is the anterior pituitary gland. So this is another name for the anterior pituitary gland, which is adrenal hypophysis. The four strictly tropic hormones are the thyroid, TSH, FSH, LH, ACTH. While the non-tropic hormone, the main difference that the non-tropic hormones, they target and stimulate non-endocrine tissue to induce effects. These non-tropic hormones are produced by the anterior pituitary gland. As an example, the prolactin and the melanocyte stimulating hormone. So if you need to understand that these kind of hormones stimulate non-endocrine tissue. For example, the prolactin stimulates the mammary gland to secrete milk. And this mammary gland is a non-endocrine tissue. So in case of growth hormone has both tropic and non-tropic effect. So that means growth hormone can stimulate endocrine gland to grow and secrete its hormone, and as well can stimulate non-endocrine gland, non-endocrine tissue to induce effects. So what's the location for different types of hormone receptor? This hormone receptor, as I said before, either in or on the surface of the cell membrane, and they are very specific and mostly for protein, peptide, and catecholamine hormones. Others are for these receptors, others exist in the cell cytoplasm, and they are primary receptor for steroid hormones. Others in the cell nucleus, and they are receptor for thyroid hormones. You need to know that the number and sensitivity of hormone receptor need to be regulated because receptor respond by interacting with the hormone. So once the hormone binds to the receptor, there is a cascade of reaction occur that trigger the effect. So this binding need to be regulated. So the number of the receptor in or on a target cell usually doesn't remain constant because it depends on the amount of the hormone. This down regulation of the receptor or decreasing the number of molecules of the receptor either on the target cell or inside the target cell can occur as a result of inactivation of some receptor molecule. So to make it clear, we are talking about the mode of action and how the endocrine signaling is regulated. So if the cell found that it doesn't need anymore the function of the hormone, it needs to inactivate this receptor in order not to accept the hormone. So one of the mechanisms, as we said, the inactivation of some of the receptors. As well, the system can inactivate some of the intracellular protein signaling molecule. Because once the hormone binds to the receptor, there are some intracellular signaling action that need to be accomplished. This signaling mechanism is controlled by intracellular signaling protein. So it's a kind of the cell interrupt this signaling by inactivating the signal inside the cells. The other mechanism for down regulation or regulation of the hormone receptor binding is the temporary sequestration of the receptor to the inside of the cell, away from the site of the action of the hormone that interact. In other words, if the cell doesn't need anymore the effect of the hormone, and the hormone is already released in the bloodstream, so it prevents its binding to the receptor by moving the receptor inside the cell, so the hormone will not find the receptor of the cell membrane. That's sequestration. The fourth method of regulation is the destruction of the receptor by lysosome. So they internalize the ribosome and destroy the receptor. All these processes for regulating the action of the hormonal action. The fifth method of regulation is to decrease the production of the receptor. In each case, the receptor downregulation decreases the target tissue responsible to hormone. And this, in all cases, this is not a bad thing, because target cell need to regulate its action in order not to be overused. Okay, so for example, if the muscle, for example, a gland needs a hormone like the thyroid gland, the thyroid gland secretes the thyroid hormone, but it needs a hormone that comes from the anterior pituitary gland. So if the action of the thyroid hormone is terminated, so the thyroid gland doesn't need any more activation from the thyroid stimulating hormone, which comes from the anterior pituitary gland. So it either do any of these 5 mechanisms. It either inactivate the receptor, it either internalize the receptor on the thyroid cell, or either destroy this receptor by lysosomes after internalization. In all cases, the thyroid cell in the thyroid gland need to prevent the interaction between the thyroid stimulating hormone and its receptor to augment any other activation of the thyroid gland. So, what is steroid hormone? As we said, it's hydrophobic and lipophilic. It acts on a nuclear receptor. It functions to change protein synthesis. This kind is called they work under the mode of action which is called direct messenger mechanism or direct gene activation, which can be described in 5 steps. The steroid hormone diffuses through blood membrane. It binds and activates nuclear receptor. This hormone receptor complex activates a gene. The gene is transcribed into messenger RNA, and the messenger RNA is translated into protein. This intracellular receptor or the nuclear receptor, so they function actually as a transcriptional activator when bound to their ligand. You can see here how the direct mechanism or the direct gene activation of steroid hormone occur. Starting with the steroid hormone coming from the extracellular fluid, it can easily penetrate inside the plasma membrane, and it binds to the nuclear receptor, which is located in the cytoplasm. Although it is located in the cytoplasm, but it is called nuclear receptor because its a function start in the nucleus. Once there is a binding between the receptor hormone complex, it is translocated through the nuclear pore inside the nucleus. Once it translocated, it binds to specific region on the DNA for the specific gene that will be transcribed into messenger RNA and then processed and then translocated. This mature messenger RNA is translocated to the cytoplasm to be translated into protein. So again, the steroid hormone diffuses through the plasma membrane, bind to the nuclear receptor. This receptor hormone complex enters the nucleus. This receptor hormone complex binds to specific region of the DNA. This binding initiates transcription of a gene, which leads to the messenger RNA that soon after that it will be translated. So you need to know that all nuclear receptors, so we're talking now about the nuclear receptor. In this example, it is the red piece here, this nuclear receptor. So you need to know that all nuclear receptors share the same domain structure. So what's a domain? Domain actually, it's a part of a protein that have a specific sub-function within the receptor. So normally the receptor have a number of domains. So you can see here that estrogen receptor, obviously, this is the receptor which will bind with estrogen, as well as the progesterone, glucocorticoid, thyroxine receptor, retinoic acid receptor. All these 5 receptors have a common general structure and common domains. So what are these domains? It needs a DNA binding domain. So actually this domain which will enable the receptor when bound to its signal, the hormone, the steroid hormone, to bind to DNA. So all these hormones share the same domain, which is the DNA binding domain. As well, it has the ligand binding domain, and this ligand binding domain, or this specific sequence of amino acids, is used to bind to the signal, which is the hormone, the steroid hormone. So the receptor has a domain for binding to the hormone, the steroid hormone, and have another domain to enable this complex, the receptor ligand complex, to bind to DNA. And the only difference is the third domain, which is the variable domain. The variable domain, it may function as activation domain. As I said as well, the DNA binding domain has a repeats of a specific structure called C4 zinc finger motifs. And these zinc finger motifs, actually, it is a specific sequence of amino acid enables any protein that have this zinc finger domain to bind to DNA. And it have, as well as I said, ligand binding domain, which is function for a repression domain in absence of a ligand. So this actually the general primary structure for all these five receptors. So how scientists knew this?
دكتور بس سؤال بس السلايد اللي قبل.
Okay, hold on. تفضلي.
It may function as a repression domain. شو معناته؟
It means that in the absence of the ligand, it prevent this nuclear receptor from translocating inside the nucleus.
Okay.
Okay, because this, if the nuclear receptor is translocated into the nucleus, it will perform the action of transcription. So it needs to be regulated. So this ligand binding domain, once there is no ligand, okay, it will ensures that this receptor will not be translocated inside the nucleus. But what if there is a ligand? So this ligand binding domain will bind to the ligand, and hence the nuclear receptor can be easily transported inside the nucleus. You got it?
Yes. Okay.
So how scientists knew these domains and how the mechanism work and everything? Okay, we'll see a very good experiment here illustrating that. So in this, to understand this experiment, so you have cells. These are cells, okay? To get familiar with this photo, these cells, you have cells without DEX, and cells with DEX. What's DEX? DEX actually, it's a kind like hormone. So that's the steroid hormone, okay? Synthetic steroid hormone for doing experiment in the lab. So you have now cells grown in the cell culture, okay? And you can see that these cells got transfected, or that's what's the meaning of transfected. That means that these cells, by a specific technique, we can introduce protein inside this cell. So scientists here used a very good protein used in research, which is the beta-galactosidase protein. What is the character? Actually, beta-galactosidase is not a hormone or anything. It's actually it's a marking protein that emit fluorescence. So they use this protein so that they can monitor where is the protein is located, because this protein can emit fluorescence, and when cells are photographed under UV, it can emit light. So this UV, when exposed to this protein, the beta-galactosidase, it emits light in the visible range or fluorescence. So you can see here from this figure, when they introduced these beta galactosidase to the cell, and in the presence or absence of the steroid hormone DEX, you can see that this protein is localized into the cytoplasm. How we knew that it is localized into the cytoplasm? Because the fluorescence appear in the cytoplasm, because the nucleus appears dark. That means there is no protein in the nucleus, it's only in the cytoplasm. So we are now sure that beta galactosidase is localized in the cytoplasm, whatever there is a steroid hormone or no steroid hormone. So that means this steroid hormone doesn't affect the localization of beta galactosidase. Again, why we use a beta galactosidase? We use a beta galactosidase as a marker to see the localization of protein. Okay, so actually this is not the experiment. So where is the experiment here? So they have done another set of experiments. They fused the beta galactosidase with the glucocorticoid receptor. As we have just said, that the glucocorticoid receptor is a nuclear receptor. So normally, if you remember from the last slide, normally these glucocorticoid receptors will stay in the cytoplasm if there is no ligand, if there is no hormone, and will be translocated in the nucleus if there is hormone. So you can see from the two photos or two figures, the top figure, you can see in the absence of DEX, this glucocorticoid is in the cytoplasm. While when we treat cells with DEX, which actually is the steroid hormone, this glucocorticoid is translocated into the nucleus. And now the nucleus is brighter than the cytoplasm. So where is this light come from? Actually, the light comes from the beta galactosidase, because we have said before that the beta galactosidase is only used for marker to visualize where the protein is. So the main aim of this figure B, I mean, it proves that in the presence of DEX, which is the steroid hormone, the nuclear receptor is translocated inside the nucleus. That means DEX bound to the glucocorticoid, and it triggers the translocation of this receptor ligand complex into the nucleus, evident by the fluorescence of the beta galactosidase inside the nucleus. Okay, let's see what happened here. So scientists wanted to know which actually the domain of the three domains, because as we said before, these nuclear receptors have majorly three domains: a DNA binding domain, a ligand binding domain, and a variable region. So they need to make sure which actually the domain that is responsible for the translocation. So they have done another set of experiments. In this time, they fused the beta galactosidase with only the ligand binding domain, So they didn't took the whole receptor. They only fused the ligand binding domain with the beta galactosidase. And you can see that they basically had the same result as figure B. So that's mean binding of DEX with the ligand binding domain is the only factor that leads to the translocation of the receptor ligand complex inside the nucleus. And it's a very good example that the ligand binding domain is the domain which controls the translocation, as we have said before. Continuing the mechanism of action, so now we have just talked about the direct mechanism. What's the meaning of direct mechanism? Direct mechanism, that means that the activation leads to direct gene activation. So, and this occurs only for steroid hormone. What about the non-steroid hormone? The non-steroid hormone, like the hydrophilic, lipophilic, and they need to act on the receptor on the plasma membrane. Examine the amines, amino acid, peptide proteins, and glycoproteins hormone. We have another mechanism, which is the second messenger mechanism. The first one is a direct messenger mechanism. In second messenger mechanism, you can briefly, it can be divided into steps. Hormone bind to plasma membrane receptor. This receptor is activated. This activated receptor, actually, it has an enzymatic feature which will affect which will activate an effector enzyme. Once this effector enzyme is activated, a second messenger is generated in target cell. Following that, there is a cascade of reaction are generated. This cascade of reaction will trigger all the responses in the triggered cell. So initially, you have here the plasma membrane. You have the receptor, for example, here the insulin receptor. In this case, the insulin receptor is inactive. Once there is insulin in the media coming from the extracellular, the insulin will bind to the receptor after dimerization. The process of dimerization needs the binding of the insulin receptor to the plasma membrane receptor. Once dimerization occurs, this receptor, the plasma membrane receptor, undergo a very peculiar phenomenon. It will do autophosphorylation, so it will phosphorylate itself. What's the importance of this phosphorylation? Actually, we need the main problem here that we need to transfer the signal from outside the cell into inside the cell. So the signal outside the cell is represented by the insulin. Once it bound to the receptor, which is dimerized, this receptor, once is autophosphorylated, now the signal is transformed to the cytoplasmic part of the receptor. So you can see that this plasma membrane receptor have two regions, or basically three regions for the receptor, for insulin receptor. You have a region that outside the cell, you have a region that inside the cell, and you have a region that transversing the plasma membrane. So once the insulin binds to the extracellular region, the signal is transmitted to the intracellular region of the receptor via the autophosphorylation. What is the importance of this autophosphorylation? Once this receptor is phosphorylized, as you see in the figure, another protein which is inside the cytoplasm will recognize this phosphorylation. And this protein is called RS1, insulin receptor substrate 1. So this protein will bind to the phosphorylated receptor. Now, the signal has been transformed or transduced from outside the cell into the inside of the cell. Let's take an example for the second messenger mechanism. It's a very good example, okay? So you have here hormone or a growth factor that bind to its receptor. Once the hormone is bound to the receptor will be active. This receptor, once it is active, it is recognized by a very specific protein called G protein. This G protein is consisted of two parts, a G alpha and G beta and gamma. Once this protein recognizes this active receptor, it will activate another protein which is called phospholipase T. This phospholipase T, from the name implies, it degrades phospholipids in the plasma membrane. So this phospholipase T will work on PIP2. What's a PIP2? It's a lipid molecule. It's a phospholipid molecule plus PIP2, phosphoinositol diphosphate. So this molecule actually function in preventing the TAPI protein. It's a type of protein, this TAPI protein. It prevents this TAPI protein from existing in the cytoplasm. So this TAPI protein is prevented, as I said, from going to the nucleus by binding to PIP2. This PIP2, once the phospholipase T is activated, it will break BIP2 into diacylglycerol and IP3 in stool triphosphate. So once this molecule, the phospholipase molecule, the phospholipid molecule is broken, the tubby protein will be released in the cytoplasm. Once it is released in the cytoplasm, it has two very important domains. One of them is a DNA binding domain, which will enable it to bind to the DNA. And the other one is a transcriptional activation domain, which will lead to a transcription of a specific gene. So basically, the initial mechanism of hormone, once bound to the receptor, it will lead ultimately to the binding of the tubby protein to the DNA to perform a transcription of a specific gene. As I said before, this tubby, in the absence of this hormone, in the absence of the hormone or the growth factor, this tubby cannot do this action because it is prevented from doing this action by being attached on the membrane by BIP2. But due to the activation of the phospholipase C, due to the presence of hormone, this BIP2 is broken down and release the tubby and can do its action. So what is tubby protein? They found that the tubby gene is expressed primarily in brain. They found that this tubby gene and the product of the tubby gene, tubby protein, is involved in the control of eating behavior and involved in obesity as well, because they found that people that are obese have high level expression of the tubby gene. How did they know that? Because they mutated the tubby gene, they have done experiment on mice, and then they mutate T gene to be hyperactive. They found that the mouse developed obesity, it was obese. So this gene, this signaling is important in regulating the obesity. Let's see another pathway again before here. So actually this mechanism, as I said before, it's not a direct mechanism. That means it's not lead to direct activation, direct gene activation. It needs a second messenger. Here you can see from the figure, the second messenger here is BIP2. Why it's a second messenger? Because BIP2 need to be broken by phospholipase C to release the tubby protein. In this case, what is the first messenger? The first messenger here is the hormone or the growth factor itself. Okay? So let's see another interesting pathway explaining the second messenger mechanism. And this pathway regulates the amount of calcium present in the cytoplasm. As we said before, it starts with the hormone, which binds to the receptor, activate a G protein. This G protein will activate as well phospholipase C. Okay. This phospholipase C, as we said before, it will work. As we said before, it will work on BIP2. This phospholipase C work on BIP2, releasing breakdown BIP2 to the diacylglycerol DAG and IP3. Let's see what happened here. Once the phospholipase 2 got activated, as we said, it worked on BIP2. This BIP2 got cleaved into two molecules. Diacylglycerol will stay in the plasma membrane, while the IP3 will be translocated to a special gate channel on the endoplasmic reticulum. As you all know, the endoplasmic reticulum in our cell represent the stores of calcium. So calcium is stored inside our cell in the endoplasmic reticulum, the major stores of calcium. Calcium is a very important molecule. It needs to be regulated efficiently. So most of the calcium is stored in our cell inside the endoplasmic reticulum, and it is prevented from the calcium is prevented from staying in the cytoplasm due to its effect. So here, once the IP3 is released, it bound to a special kind of a gate channel for calcium gated channel, which upon triggering, it will release calcium from the endoplasmic reticulum to the cytoplasm. So that's why this channel, this gated channel, is named IB3 gated channel, IB3 gated calcium channel. So that's means that this calcium channel only needs IP3 coming from the plasma membrane to open the channel. Once the IP3 bound to this channel, it will release calcium in the cytoplasm. What happens when this calcium released in the cytoplasm? This calcium will will activate a very important protein kinase called the protein kinase C. This protein kinase C will be bound to the diacyl glycerol for further phosphorylation of substrate. So this ensures the continuity of the cascade and the metabolic responses inside the cell. So here you can see that signaling occurs in different ways, but the cell needs to interpret this signaling to do the appropriate response. Actually, in most time, the signal receives a lot of multiple signals. And for regulation, it's very important to regulate the signal coming to the nucleus. Otherwise, malfunctioning will occur. So you can see here, in order to transcribe a gene, this needs the coordination between 3 different signals. And this is a very important mode of regulation, this like a general mechanism, okay? This is a very important mode of regulation to ensure that the gene that will be transcribed is the correct gene. So here, as a general mechanism, you can see that this hypothetical gene need to be activated and transcribed under the influence of the 3 different signals, which require 3 different messengers, which require 3 different receptors. Each receptor will have its own pathway, and all of them, the nucleus will interpret this 3 signaling pathway to do a transcription for the specific gene. In this case, you can see that the second messenger differs for each signaling pathway. The third effector, so here, the third effector, you can see it's the PKC, as we can see here, okay? Here you can see that the calcium is a second messenger, while calcium NK, calmodulin kinase, is another effector. In other words, all these 3 signals had to be coordinated to lead to one common signal, which is the transcription of a single gene. So it's a mode of careful regulation of a transcription of gene to ensure that the correct gene is transcribed. Continuing the hormones and homeostasis regulation. So hormone transmit information about the status of one organ to another. So they need to regulate the corrective action to maintain homeostasis. For example, if the glucose is elevated in blood signal, pancreas trigger to release insulin. This insulin travels through the circulation, leading to the signaling to liver and fat cells to increase their permeability to glucose. So why they increase the permeability of glucose? To have the glucose stored inside this liver as a glycogen, aiming to decrease the blood sugar level. To be effective, this hormone should not be degraded too quickly or too slowly. The hormones have a very powerful effect. It needs to be released at the correct concentration, because any disturbance of any hormone in our body, it will affect a lot of function. So that's why it needs to be not degraded too quickly or too slowly. Why? If the hormone is degraded too quickly, it will not arrive at the target site. That means if it's degraded while it's moving in the bloodstream, it will not arrive at the target site. But what if it's degraded too slow? The information conveyed will be obsolete and may evoke an inappropriate response. That means if the hormone is degraded too slow, that means it will stick, it will go to the target cell, and maybe the response needed is already passed. So it doesn't need the hormone anymore. Okay, and this may lead to any other response that might affect the target cell. So you need to understand that different hormones have varying half-lives according to its action in the circulation. Some hormones exist for a second in the bloodstream, minutes, even hours. It depends on how the mechanism of action of this hormone. Actually, this half-life in the circulation of the blood circulation, it depends in part on the distance that the hormone will travel, the signal will travel, and the nature of the information that it needs. This means a thyroid hormone, when travel to the blood, in the blood, it's totally different than the insulin hormone because the distance is different for traveling, and as well the message that will be conveyed will be different. Okay, and this concludes our second